Okay, so now moving swiftly on, we now introduce Sir John Byrne, who's Professor of Clinical Genetics at Newcastle, and not only involved as a clinical geneticist, but also some of you will know him for his development of quantum medics and its amazing kit. Sir John.
Thank you. Ladies and gentlemen, it's an honour to speak after this stellar cast. And I thought I'd choose a slightly challenging title, so at least you'd stick around and not go for an early coffee. But this is not a negative talk. It is, however, a realistic talk. But just to follow on the Minister's analogy with space, it's worth remembering that this is one of 3 massive revolutions that have transformed my life and your lives in recent years. Not only do we have genomics hitting us, but we've also got those geostationary satellites that allow us to talk to each other constantly with our mobile phones, and through those mobile phones have access to all the world's knowledge in every library on the planet. I mean, imagining that when I was a medical student would have been very challenging. Like Sally, I've been in this game for 4.5 decades, and in fact I thought I'd start with this certificate because it was given to me by Victor McCusick, who I went to work for as a medical student in the Mayo Clinic in Baltimore. We went to the Jackson lab to do our summer school, and I got my certificate in medical genetics from Victor. That's what I looked like. I don't think I knew I was having my picture taken there, and I'm quite certain my wife wished I hadn't taken that one. Don't tell her I showed you it. We actually had nothing to live on, so we ate lobsters, clams, and stole sticky buns from the conference, because we had no money. But in the background there is Victor McCusick, who later had me join his editorial board on Omium, the online medical textbook. He's the one on your right with the bad dress sense. So Victor was probably the world's greatest clinical geneticist and only died recently, and he inspired me to do this. But the reason I put the picture up is he also chose the term genomics for a new journal, and that's what really kicked off the term. So really I'm from the pre-genomic era, which makes me very old indeed. In fact, I chose to get into genetics like Sally. In fact, the double helix inspired me, but what really inspired me was at the age of 17 being taken by our biology teacher to hear a lecture on the genetic code. People tend to think about the double helix, forget the genetic code is an equally brilliant breakthrough, mostly by British scientists, working out how you take 4 letters, in this case the 4 letters of RNA, and explain all of human life. And it was apparent that in order to account for all the amino acids and a bit of grammar, you needed to look at them in sets of 3 in order to have enough combinations to cover all the bases. So for example, you take the letters G, U, and C, and you get a valine. But the fact is, because you only got a couple of dozen requirements, you've got 64 combinations, it's a degenerate code. So in fact, you could take GU and anything, and you still get a valine. And therein lies one of the problems we have when we start whole genome sequencing. You could change millions of letters and make no difference whatsoever. Conversely, change the U to a C, and you switch a valine to an alanine, and that can be massively disruptive to the function of the protein. So working out which letter matters and which doesn't as we move into this huge new data world is going to be a major challenge for us. And as you've already heard, informatics, I don't need to tell this audience, is where we really need to make major progress. So as a clinical geneticist, this is the sort of family that I've been dealing with all my life. I've been a consultant now since 1984, which makes me feel very old. And this is Lauren Trudy. I saw Trudy when she was a little girl, diagnosed as having Noonan syndrome, and then had to think with her whether she wanted to take the chance of having a child or not, because it was a 50-50 chance. She decided to go for it. Lauren, in fact, inherited the gene and had a very major heart defect, which we managed to successfully correct. The problem was I couldn't find the cause. We spent years sending DNA from this family all over the world, and we didn't know which gene caused it. But we could still make a big difference to their clinical care. Not only do we diagnose and give prognosis and therapy, but we can also discuss why it happened, will it happen again, will it be as bad, and are there any tests? And of course now we have the tests. In fact, we found it was a gene called BRAF that was mutated in this family with the help of Dutch and Japanese teams, and it took us 10 years. About 10 years ago, I was at a conference in Manchester where we were discussing whether we could decide which of the increasing number of genes that we should test for Noonan syndrome, and I said, why not just shoot them all? So we actually set up an assay with St George's. We have a spin-out company called New Gene owned by our hospital, and now for £970 you can test all the genes that cause Noonan syndrome in a single shot, and BRAF is there on the right hand side there. So that was a big step forward. It meant that instead of waiting 10 years and begging researchers to do it, we could order a test and get the answer. And then, of course, that opens up possibilities of things like pre-implantation diagnosis in order to avoid having a child affected by these quite serious diseases. We've gone on from there to exome capture. So this is some work from my partner, Patrick Chinnery, and he's a world expert on mitochondrial diseases. And this is a paper he published where he showed that some of those vanishingly rare syndromes you've just seen described, if we pull out the sort of 1.5% of the genome that is the coding segments of genes, as John McGrath, the dermatologist in London, said earlier this week, who were at the conference we were speaking at, he said it's a bit like watching Match of the Day instead of watching all the football games in the country. And so you just see all the goals. And so you can basically pull out that small percentage of the genome. And this is a very attractive proposition in clinical practice because you go to the key genes and look for spelling changes that we can understand the meaning of. And we get about 2/3 of the cases explained by that approach. And the DDD project in Cambridge, which I had the pleasure to sit on the advisory team for, has had major success in using whole exome sequencing to look at families to find diagnoses. So why do we go to whole genome? Because whole genome's actually got more information for us. But before we do, just to think about the challenge, this is Patrick Chinnery, who took over from me as head of the Institute in Newcastle, and we were hosting at our science centre the Wellcome Trust display on genomics, and this is what your genome would look like if you printed it out. That big book in the middle there would be what my X chromosome looked like. If you actually zoom in on it, in fact, if you look very, very close, you have to have very good eyesight to read the letters in order to be able to print the whole X chromosome in that book. And that's, I think, page 835 of 1,163 pages for your X chromosome. So that's the sort of quantity of data we're dealing with. I show you this because I sit on the board of NHS England now, and I'm proud to say we are the world's biggest purchaser of fax machines. Now imagine what would happen if we decided to fax this data to each other. You'd probably spend the whole of your life doing that job. So we've got to actually deal with a switch from this paper-based fax machine world into the digital world that Sir John described before me. We've got to get the informatics of the NHS working if we're going to have any success. I'm proud to say that among the 1.3 million people the NHS employs, I imagine that tens of thousands of them this morning have already written the letters DNA on someone's medical record. Sadly, they use it to mean did not attend. And in fact, when you actually say to colleagues, what does DNA stand for? They go, in fact, we haven't got there yet. We've got a major educational challenge. And one of the things that we're doing with the Genomics England and with the response to that is we've now set up master's courses in 6 centres, including our own. In fact, just yesterday I gave the opening lecture to our second course, where we're bringing in clinicians from every branch of medicine to teach them the basics of genetics so that they can actually at least understand the reports that we're going to be sending out to them in the near future. So we shouldn't be, we've heard 3 enthusiastic supportive statements, but could genomics fall because it doesn't do what it says on the tin? And there is an element of this. We've got to bear in mind that when we do a whole genome sequence, we've got error rates to deal with the technology. We've got inadequate depth of coverage. So if you're using the Illumina technique, it's a bit like a Monet painting. It's lots of little dots. And it's possible to miss segments unless you have at least 30-fold coverage. There's huge segments of the genome which are replicated. And currently, whole genome sequencing picks up about 85% of those. But that's a big miss if the other 15% was actually the cause of the disease. And then you've already heard Viv mention the issue of epigenetics, imprinting, chemical changes in your DNA, which will actually profoundly change the way it works and is partly influenced by our lifestyle. We don't pick that up yet. So in truth, when we do a whole genome sequence, we actually spell it wrong. There is actually still holes in this process. And so we shouldn't just assume if you've had your whole genome sequence, there you are, we know everything. So this technological challenge is still real. We've got so far half a dozen techniques. Sanger sequencing, the one we did for the whole Human Genome Project, is great, but it's really slow and laborious. But that reads one letter at a time. All these other techniques are different approaches, and at the moment none of them give exactly the same answer. And so we've got to try and squeeze that technological variation out of the system. Illumina are clearly the team leaders at the moment, and that's why Genomics England opted to go with them in terms of the Genome Project. But new technologies are coming along, and I'll mention one in a minute. But the point is that what we're finding from all these techniques is that each one of us got about 3 million variants that we've got to sift through to work out what works and what doesn't. You've already heard about the Human Genome Strategy Group report that led to Genomics England, which we've heard about from John. It's worth remembering that this was published at a time of considerable austerity. In fact, that's why we only got a half a photograph for the front. And so, I mean, basically, we've got to put this into the NHS at a time when our finances are under intense pressure. So we've got to work out how to do this realistically. We can't simply just pick it up and run with it, because we are, as you know, trying to actually cut costs. And while it's true that genomics will cut costs long term, hospitals trying to balance their budgets will need help to get this into play. This is an example of why we don't just stick with exomes. These are 3 young people who, 2 brothers from Darlington and a little girl, Sian, from my hometown of Bishop Auckland, who I met back in the early 90s. And with my colleagues, we described them as having a new syndrome. One of the features that struck us about them was that they had blockage of their noses, coronary atresia it's called. They couldn't breathe through their nose. And they had a very particular pattern of eye, ear and face malformations, but normal intellect. And I spent years trying to work out what this was. We wrote a paper about it back in the early 90s. A family was found in Alaska. They did exome sequencing. They couldn't find the cause. But then Bill Newman in Manchester said he found a family, said, let's have another go. We teamed up with German colleagues to do this. And this was the paper that came out. It's the only time I've been an author on a paper where I was also in the title as well, because it's now called Byrne-McEwen syndrome. But the reason I put this up is that this was an international collaboration that pulled together these cases and do whole genome sequencing. And what we found was the reason that these children had their problem was that they had lost one copy of a gene on chromosome 18, and they had a deletion in the promoter of the other copy. They were so-called compound heterozygotes. And that promoter deletion knocked the gene down. It didn't stop it working, it just slowed it down. The fact is, this is a gene that's critical to splicing. It's what assembles your genes when the fragments are copied off the DNA. If you completely lost that gene, it would kill you because you couldn't assemble any gene. But if you've lost one copy and you've got one copy not working very well, you can kind of just about manage to splice your genes together, but it causes malformations. So in other words, as we start to understand more about how genes work, we're going to need a whole genome sequencing in order to pick up this subtlety, because it's the promoters and the non-coding RNAs between the coding segments that are the fingers on the strings of the instruments. They're the bits of the genome that will regulate gene function. And many of the diseases that we are struggling with are actually not because of the coding segments, but because of the bits in between the coding segments that make them work. So I'd like to go back to that original example I gave you of the whole of the genetic code. These are 2 sisters who I saw recently, Christine and Kathleen, who gave me permission to use their picture. And Kathleen had breast cancer. They had a strong family history of breast cancer. And my colleague genetically tested them, found that she had a mutation in BRCA1, and went on to test her sister as well, who also had that mutation. But our colleagues in the lab said this is a variant of uncertain significance. Or if one of my Nazials here speak tomorrow says a very unhelpful statement. Because a VUS is constantly being published. I mean, there are tens of thousands of variants of uncertain significance in BRCA1 and BRCA2. And so our counsellor said, what should I say to this family? And I looked it up, a quick look on Google on my phone in the corridor. And it turns out that there was a paper published in 2007 by Doug Easton, one of the world leaders, where this mutation was seen alongside another mutation in BRCA1 in a patient in America. Now, you're not supposed to be able to survive losing both copies from birth, so they assumed this one was not pathogenic. However, when Susan Domchek went and found that patient, it turned out that lady had microcephaly, short stature, extreme drug toxicity. She was the one survivor of having 2 faulty copies of the gene. This is a pathogenic variant, and in fact, when you look at it across all species, that valine is present in every species on the planet that has a BRCA1, and all of them have BRCA1 because it's a DNA repair gene. Now, for it to be conserved so consistently is clear evidence it's an important amino acid. Changing that valine to an alanine is disruptive. So we can now tell that family, and indeed I did tell that family, that in fact this is pathogenic, and on the strength of that, Christine also had bilateral mastectomies to avoid getting breast cancer. We give 10 million prescriptions a year of warfarin, and about 5% of us are sensitive, but we don't test people. We need to test tumours in order to decide what to do with them today. We need to test people for infectious diseases like malaria. I'm going to skip this slide but just to say that we don't genotype for warfarin, even though we've shown by a randomised controlled trial that that would avoid dangerous bleeds and get people up to the right dose more quickly. But also, infectious diseases needs genotyping fast. TB, in 2005, the red places were the world's TB resistance. This is the drug-resistant TB now across the planet. And Britain has more TB than the United States. We've got a major problem with this. We need devices that will give us a diagnostic tool. So about 10 years ago, my partner, Jonathan Mahalloran, was sitting next to Charlie Lieber in Harvard, and Charlie invented the nanowire. And Jonathan realized we could actually stick DNA on nanowires, and when they met their matching partner, the wire would detect it. And so we decided to build a device. The minister very kindly posed when we went to the signing of the 100,000 genomes contract with our device. And this is the little cassette, but we've re-engineered the whole process. We can extract DNA in 2 minutes, we can do PCR in 9 minutes, and then you binded your PCR products onto a chip the size of your thumbnail, which will give you an answer, the whole thing inside 20 minutes. And I can today, since the minister's presenting his breakthrough, show you the QPOC. And we are hopefully going to have this on the street this year. We're working with international partners now. And basically you'll take a sample, whether it's a spit sample, for example, for TB, or a warfarin test, pop it in, have a cup of coffee, press the button to go, and it will do the test for you and give you a result in 20 minutes. So I'm hoping to see that actually on the street. There's the DNA test there for one of the warfarin markers. That's the non-complementary DNA, that's the blanks. Very clean results. And so the device is now virtually ready to fly. And what we're hoping to do is see these in the pocket of clinicians around the world. This is not instead of whole genome sequencing, it's taking the knowledge gained from whole genome sequencing out into clinical practice so that people see genomics as part of their routine practice. So in summary, and thanks to the 50 members of our company now, in summary, DNA analysis is going to cut across all medical disciplines. You've all got that point. We can use panels, we can use exomes, and we can use genomes. They all can work together, although increasingly we'll move towards whole genomes and then use them for virtual exomes, etc. We must upgrade our informatics. This is going to be what stops us in the NHS in particular. But I think point-of-care genetic testing will have a place because it's fast, it's cheap, and you can throw it away. You can do the test you need an answer to and then discard it. So I think that the answer to my title at the beginning was that this is going to be the rise and fall of genomics, not because genomics will fail, but because it will be so successful that in the very near future, genomic medicine will just equal medicine. Thank you for your attention.
Excellent stuff. Always inspiring to hear you. Questions from you? Hands up for questions? Well, actually, I've got a question for you while people are thinking about it, because the Centre for Life has been very, very successful. You mentioned 40,000 children a year going through it. I mean, I think this is going to be one of the recurring themes of this event, that we need to get out there and to tell people about genomics, not only because we want them to understand what we're doing, but also to increase people's confidence. And of course, for our own purposes, we'd like lots more informaticians. What's your experience when you're out there with kids, how they react to all of this?
I mean, the kids love it. Just as Sally and I were inspired by double helices and things, the kids they get this, and so I give lots of. I gave a festive lecture to a couple of hundred year 10s, and they were absolutely enthralled by it. I mean, I think we really we can win them over, but I think that the biggest challenge, actually, in a way, is not the kids. It's the staff that we already have. The people who sort of say, I can't do that. It's like, I don't do maths, so you get people say, I can't do maths. You say, what's 2 and 2? I'm sorry, I don't do maths. So you get the same reaction from a lot of clinicians. Genetics, that's something that is clever. Just send it to the geneticist. So we've got to get everybody. That's why I think our point of care device can have a big impact on people's psychology. And that's why, and Bibb, in fact, was one of our early supporters, and many people have been saying to Bibb, she made a little film, if you haven't seen it, you must watch it, it's on YouTube, about genomics in medicine, and she showed our device in it, and people have been saying, don't be stupid, you can't do genetic testing on the spot. And she says, you can, I've seen it, and it's true, you can. So we've got to kind of demystify this, and I think, and in fact, we've now been granted patents for sequencing on this technology, so in fact, I think as John said, we're going to get sequencing down to a very low price. I mean, in the very near future, it will just become part of routine care.
Hi, good morning. I'm Michelle Krishnan, a clinical research fellow at King's College in London. Hi. Just thinking about diseases in terms of gene sets and pathways that work together. And so, for example, I work researching newborn babies and thinking about this point of care testing is obviously very, very relevant in that context. And I just wondered how many variants would you be able to put on your device if you wanted to do
that's a very good question. So in the initial stages, we're actually, because nanowires, to use them to good effect, standardizing them is still delicate. So we're going to use a fluorescent detector in the very first devices, and we'll just be using 3 or 4 tests, because for many situations like warfarin, you only need 3 snips. But actually, the attraction of nanowires is we could get about 1,500 features on a square centimetre chip. So in fact, for example, all the common variants of cystic fibrosis, for example, could go on. But even now, one of the things I'm hoping to do soon is there's a mitochondrial mutation you're probably aware of for gentamicin sensitivity. About 1 in 500 babies is sensitive to gentamicin. 1 dose will make them stone deaf. And yet we don't test for it because it's too much hassle. So when babies get infections, they get given gentamicin, and then we just shrug and say, oh, really sorry, your baby's gone deaf. So, I think we need that test out there. And there are lots of examples like that where we could, even just with a single snip, actually start making an impact on clinical management. But yeah, I agree. We need to get this into primary care, into neonatal care, because that's where you need instant answers.
Quick question from you, and I have to know who this is. This is Angela Douglas, clinical lead at the Northwest Coast Genomic Medicine Centre.
Thank you, Vic. John, how long do you think it'll be? I mean, we've already started to mainstream genetics and genomics to some of our clinical champions out there in medical practice. How long do you think it'll be before our GPs start to recognise those patients when they hit in primary care and start to get them tested at that stage so that we're diagnosing these patients earlier? I mean, it would be fantastic if these patients could then turn up at secondary care with their test already done.
Well, I think that to some extent the GPs are quite reasonably trying to defend us against deluge, because our resources are limited and we're not training enough counsellors, we're not training enough people in genetics to help pick up that referral. I think I'm an optimist in life, always have been. I think that once you start getting some real payback, then the GPs will latch onto this very quickly. And I think that's why these sorts of simple tests are what they need, the yes/no question answers to questions.
Because GPs are, though, incredibly well placed for something like familial hypercholesterolemia.
Absolutely.
I mean, not only are they the ones who are seeing very high cholesterol levels when people pitch up, but they also probably know the families, and actually for them to do the extended family testing is really exactly where it should be done.
Absolutely, yeah. I mean, they should go back to being family practitioners, from being general practitioners, and actually looking after the extended family. And I think this will be one of the vehicles for them doing that. So the answer is, it's going to be tough. And I have to say, as a board member of NHS England, you know, we can't just catch this ball without help. It will take us a few years to actually build up the speed to absorb this impact. But there is huge enthusiasm for this, and I think once people see payback, clinicians very rapidly learn the tricks. At the moment they can't see the benefits, but they soon will.
And of course we mustn't forget about patient pull as well, because
I think
actually
that's important,
the
yeah.
demand for this type of testing, I mean, if you talk to people who have that awful INR testing and they have to trudge off to hospitals and it takes them forever, and actually for them to be able to do
Well, yeah,
it.
I mean, we sometimes take up to 8 weeks to get people onto the right warfarin dose. You could do a blood test and put them straight onto the right warfarin dose. Imagine you bought your shoes that way. We'll give you a pair of size 6s. Come back next week. If this sore, we'll try a 7. And then next week we'll try an 8. I mean, it's just ridiculous.
I thought that was actually the business model of ASOS, but anyway, never mind. Thank you very much indeed, Sir John.